something, you have to read it and proofread it, and you're laboring over words and concepts, and it seems like the closer you get, you know, the harder it is to get it done. It's kind of like putting the finishing on your house or something. It seems like it never gets done. So anyway, we're getting there, but it was a fairly simple weekend for myself. So let's talk about science. We're in a chapter where we have about 6 parts to it. We're winding down to the last few parts. Some things we've already talked about, maybe earlier in some of the lectures, and some things may be new to you, so that's always kind of fun to always maybe learn 2 or 3 new things and put it into this scheme of how you're thinking about life, because that's really the overall reason you have a major in any type of discipline, is you're developing a scheme of how the world works. It's just one of possibly many schemes, and as you read the news, there's people with all kinds of different ideas on how things work. But we're just building detail into this structure that you have kind of this 3-dimensional landscape going on in your head. So that's what makes learning hard, because you're always shifting your scheme, you're always trying to put new information into that, and sometimes that's unsettling. So we'll try to make it as smooth as we can, but it's even harder when we start looking at things that we can't see. So we use a lot of pictures with that. We worked from the cell membrane, and we went out a little bit to the cell wall. We started talking about the difference between the gram-negative and the gram-positive. And as we start thinking about things, if you had to tell me the difference between a gram-positive and a gram-negative bacteria, what's something you could say? Tell me some differences. There's several.
In gram-positives, the peptidoglycan is like way thicker.
Way thicker, yes. That'd be metrically way thicker. Okay, no, perfect answer on that. So we have many layers of peptidoglycan. Something else? Mycolic?
Am I saying that right?
Mycolic, it doesn't matter, you know, tomato. But I think you're saying it just fine. That's in the acid-fast bacteria. So that'd be like with the mycobacteria. Maybe I wasn't clear on that. Unless you're saying teichoic acid.
Yeah.
Teichoic. Then yes. Okay, good. I didn't quite hear you then. Here you're right. Teichoic acid is found integrated into the cell wall, and we didn't worry about the structure, but what's the so what factor on that? And what does it do to the cell that might be interesting for us?
Brings in
Negative charge. There you go. That's good. Okay, a few other things. You guys are doing good. You first, you second. Good.
And gram-negative, the peptidoglycan is in between the outer and the inner membrane.
Yeah, so we have a lot of information here. Now we have an outer membrane, and we have a little bit more information on the peptidoglycan. Was that, you got something else? Well, there's more. Which one stains purple? Positive, and we have some kind of idea why that works. I won't go through that again, but there should be something in your mind like, oh yes, you know, there's purple and something about a Morgan and stuff on that. Anything else come to mind? Yeah.
Doesn't like gram-negative have the paraplasmic, not really space, but
We call it the paraplasmic matrix.
yeah.
Yeah, that's good, and it's actually a very vital area. We'll see it in metabolism. We don't talk a lot about enzyme breakdown in this class. It's usually we get into that a lot more in advanced, but we know that something about that goes on there. What about this outer membrane though? One last thing, tell me a little more about the outer membrane. I'm from outer space, I never heard of this before. Tell me more about your waves.
The outer membrane has a lot of like proteins and it's highly specialized, so bacteria that are gram-negative can go places and be in certain environments that typically gram-positives can't.
Yeah, so he's very good, and he's talking about now adaptation, application of the outer membrane. So that's important, because that builds on our idea of diversity as we talk about that. And hopefully you're making notes on that as you go. I don't know how I can be any more obvious about pay attention to that theme. Something else with the outer membrane though, I said one more thing, but I'm looking for a little bit more, go ahead.
There's glycocalyx in the outer membrane. It's
Yeah,
what's-
attached to the outer membrane, as we work ourselves out, we're gonna come up to the glycocalyx next, so that's associated, and that's one of those optionals. Some things have, some gram nags have a lot, some, it's not as big of a deal. You know, we'll see in lab, Klebsiella pneumonia, it's just a slimy old bugger, and it's one of the reasons it attaches so well and does pneumonia so well. Well, what I was looking for, I don't want to belabor this too much, I was also looking for that lipopolysaccharide fraction. That is, what makes the inner and outer membranes different on that structural level. And we know that there's kind of a lipid moiety to it. There's a specific area, and then we have this kind of heterogeneous, you know, outer sugar area, and you go like, well, why can't you tell me more about it? Well, there's 35,000 different species. You know, let's just say even 2/3 of them are gram nags. It gets a little hard to explain that part, but it does become important there. Well, that's a good place to jump into our next thing, and we already brought up glycocalyx. This calyx is, think of it as a shell or something that surrounds something. The calyx on a flower inflorescence is something that is holding up the petals and the sex organs of the plant, the angiosperm. So here we have a calyx that surrounds the cell. And the glyco part means, to remind you that it's sugar, so these are polysaccharide strings. I don't know if layers is the best word for it, but so it's not layered like the peptidoglycan is layered. It's more like there's these strands interwoven into each other. And this is just a stain showing the cell itself, and then they came around with a negative stain around it, and the stain does not permeate the capsule too much. Now, this is just a regular aqueous stain. There's nothing that special about it. And I'm not sure, but oftentimes we use India ink, something that simple. But you can see that you get several pieces of information from this, right? First, you see, oh, wow, that is much bigger than the cell. It really changes that idea of particle. We said before, maybe look in your notes, we said, well, that can be important because that can affect how fast the cell might gravitate out of solution. You know, and these things are small. They don't just like plop to the bottom like gravel when you dump it into water. But if you even put kind of a balloony sugar structure on the outside, it even affects their buoyancy differently. They do not, though, here they do not affect their own buoyancy actively. This is a passive thing. And we see that it's big compared to the cell we talked about. And the other idea we get from this is, well, something about that capsule doesn't let the stain get in there too fast. It seems like, or doesn't hang on to the stain when we wash it out. And that's that real sugar matrix. It's true, it's negatively charged. Do I have that up there? Well, if it's not up there, yeah, there it is. Negatively charged. That's what they'll call this a mucopolysaccharide matrix. And it does have a negative charge to it. So, and this is where we started to talk about some of that bridging a while back with the divalent cations. If we have, so if this is the cell, and here's the outer membrane, and here's the cell membrane. Yes, this is very simple, but I'm trying to stay and keep things going. If we have this matrix out here, sometimes or oftentimes you'll see calcium. If there's taking the negative charge here, making a bridge here and that. Now it won't saturate, it won't become like rock hard, but it tends to occur in there. Alan Decco had an interesting thesis. He said that even things like iron will come and make double bridges in there and even affect it differently. So he was very interested in how bacteria react in the environment, and his whole thesis and all his work was really on the capsule and the extra, what they call the extracellular layer, because it's extra outside of. And there's quite a literature on that now. In the ecology world, maybe I'll be right with you on that.
Sorry.
Let me finish the thought, I'm old, I forget. In ecology, you might learn about how aquatic insects eat leaves and so forth, and they have a theory called the peanut butter and cracker theory of nutrition, and that is the leaf, because it has cellulose and lignin, is kind of the cracker. There's a little bit of nutrition there, not a lot. Alan Decco and a lot of other people said, well, you know, when you coat these leaves with all the sugar and protein from the microbes, that's really where a lot of nutrition is. And that's kind of the peanut butter idea, except peanut butter high in fats. There's not a lot of fats here. So, but that's as far as that analogy goes. Maybe some of you can talk to Dr. McEwen about it. I'm pretty sure she'd be aware of that idea. It's fairly common and dates about 15 or so years now. People have been using that analogy, but it's another place where microbes and this matrix become important in ecology and adaptation and disease.
So the bacteria that eats the iron, would their capsules then use iron more commonly than the calcium?
Yeah, now this isn't necessarily eat or metabolize. This is
Well,
more
because
it's associated. And Decco's theory was that as a biofilm, back to biofilms.
yeah,
So in a biofilm,
actually I don't.
so in a biofilm, let's just change this for a second. So let's just say this is our leaf, and we have our biofilm matrix here. So there's all kinds of different species here. And we know it's kind of like a mushroom cloud now and so forth. But he's saying that biofilms will form like succession. We'll have cells come in, then they'll start to grow, and it'll age, just like on your teeth. If you don't brush your teeth every day, it becomes pretty scummy, and at some point dental caries, and then you have real problems. But he's saying that these mushroom clouds might start to compress, and these things might start, it might initiate the sloughing off of the biofilm, because the iron, as it ages, grabs more and more iron as it ages. Because iron in oxidized environments isn't that plentiful. It's really more plentiful, iron is more plentiful when you're in a reduced type of situation. So that's the idea. So yeah, it'll help with the compression. It could change the overall operation on the community level, is what he was saying. Okay. Good. All right, well, let's move on. This is just showing kind of another view of where we're at in the cell. It's kind of a crazy looking drawing, but we know the nucleoid, we know there's ribosomes in that cell wall. We're talking about the capsule being to the outside. So what I like about this is it helps to give kind of a scale. You know, if we sliced it in half, but realize that it's a calyx. It's like the hard coating, or not, well, it's like a coating around a peanut or something, I suppose. It surrounds the whole cell if they have it. And I like this analogy to think about. I've talked about before, but I like this idea of something really, really fibrous. You know, I think everybody's seen at least cotton candy, if not eaten it. Anyone eat cotton candy?
Like
3 people have eaten cotton candy.
Yes, yes.
Anyone's, oh, okay, more of you, see, I'm a cotton candy eater, but I don't want you to eat it. Okay, it's okay with me. Your secret's safe with me. So, but anyway, so you get the idea. This real fibrous nature. Now just switch the bonds in there. Don't make it so soluble so it melts in your mouth. Think about as really fine strings, and then think about putting water into that. It's going to be like this huge sponge, around the cell. 95% in general of the capsule is hydrated. It holds water. If you were to dry it down, you'd lose 95% of the weight from water. Okay, whether that water happens to be around your lung or in a stream, whatever that case may be. So we talked about another function for the capsule is to act like a water well, as a reservoir. That is, it's one nice thing if you can all just mobile around, get transported to the places you want to be. We're real lucky that way, but you're a bacteria and you're stuck somewhere in a biofilm, in a tissue, or whatever happens, and it starts to desiccate, this buys you a little more time to put away nutrients, as we'll talk about in this lecture, maybe time to set up an alternate type of lifestyle, life form, assist or something, or to even encapsulate. Some of these things just go, just resist all kinds of environmental conditions. It's fairly amazing. Okay, let's see. So, okay, well, I think that's enough on capsules. We're going to move on to little bitty fibers. Okay, and here we're going to talk about pili and fimbriae. Okay, pilus and the fimbriae. Okay, the fimbriae are filamentous protein structures, and not to be confused with the flagella. We'll get to that more, but the flagella is proteinaceous, but it has a lot more complex morphology, we'll find out. These are basically protein strings stuck out of the cells. Again, not all cells have fimbriae. Okay, a lot of them do. Okay, what used for? In general, the fimbriae are used to stick on surfaces, right here. It's another tool in the toolkit if you're a bacterium. Okay, not terribly, a lot to say about that. Whoops, let's go the other way. If we can, come on. Okay, the other one, the type of thing, it's a shorter type of structure called the pilus, plural, pili. Okay, and the pili, the fimbriae are shorter, the pilus is longer, and we, for the attachment kind of thing that the pili do, it's kind of like the fimbriae. We don't make a lot of distinction on that. It's just a matter of size. But where people have put a lot of time and resources into discovering things is to look at the pilus as an exchange of genetic information. So it's called a conjugative, or to conjugate pilus. Okay, conjugation here, a little different than what you may have learned about paramecia. That's a different type of conjugation. If you didn't, don't even worry about it. Conjugation here is simply exchanging genetic material alongside the pilus, which is hollow. Generally, a plasmid can unravel, start making its way down the pilus, and make it to another organism. Antibiotic resistance can be transferred between organisms. If one doesn't have it, another organism can do it, for instance. That's probably the most famous one. I mean, this is a fairly unique picture. Okay, think about it in nature. You have something this thin, I mean, a little protein strand. All you need is just a touch, a wave in the solution or something, and it becomes broken. A lot of conjugation processes are interrupted, and it's only one of several ways of exchanging genetic information, but it's kind of interesting. We talk about, and we'll do it again, but we talk about vertical movement of genetic information and horizontal movement of genetic information. So you kind of think of it as a two-way dimension. Okay, when mommy and daddy had you at some point, that's vertical. Your genes were passed on to the next generation, and at some point, some of you will send your genes on. So that's that lineage. Okay, horizontal genetic information is exchanging within the same generation. Okay, so I want blonde hair, or give me blonde hair, or whatever. I would, we would be able to exchange that information. We can't. Okay, or blue eyes, or whatever. I mean, we have what you know that. But with bacteria, it's an option. You can exchange genes within the same generation, and that really drives people nuts when they're trying to really think of classic phylogenetics and inheritance. So the pilus is part of that story. We'll learn about transformation and a few other mechanisms, but conjugation comes up here. Okay, I want to talk next about some of the many inclusion bodies. Okay, we tend to think that bacteria are pretty much like that opening shot. We know there's ribosomes in there, there's cytoplasm, a nucleoid, maybe a few other things, a bunch of enzymes, some DNA, so forth. But we tend to stop there. This is interesting. One of the types of storage polymers is polybeta hydroxybutyric acid. Okay, this is a storage compound. You don't have to know the structure, just that, you know, it's kind of looks like it sounds there. So, and another thing that can be stored, someone mentioned this the other day, is glycogen. We generally don't think of glycogen with unicellular organisms as much, but it comes up here and in the algae once in a while. What's possible, as opposed, when we see these kind of, if we stain something, now this is an electron micrograph, so if we stain this or dehydrate, we see big things in here, we tend to, from our experience, say, oh, those are vacuoles. You know, I saw those in paramecium in Bio 141 or something, right? That's what usually comes to mind, and we don't talk about vacuoles too much with the prokaryotes. It doesn't show up too much. This is really a storage of one or two of the, well, this is PHB. The storage of a lipid, it's not going to pick up stain as well. Stains are usually basic or acidic, and they tend to bond on to things like nucleic acids. So it's not bonding here very well, but we can see this organism is picking up, has some storage materials. If you look at the life of a bacterial cell, a lot of times, most of the ones that you're going to see on the microscope are growing, dividing, growing, dividing. Every once in a while, so you'll see elongation, and you're like, oh, I think it's a rod because it's so long, but it's actually, you know, getting ready to do binary fission. We don't see a lot of detail. Okay, this gets kind of messed, and most of the time, bacteria don't sit in this state. They don't want to carry on in a bunch of extra mass. But we used to measure these things as stress. You stress a bacteria, so it starts going, you know, I'm going to kick in this pathway and start storing things because I think something bad is coming my way. Okay, I mean, they think that bacteria thought. That's how they talk. You know, so. Anyway, you don't always see it in all the bacteria. Sometimes you have to induce it if you're studying them. When you look in nature, it's even harder because there's so many different things out there. Polyphosphates can also accumulate. This is really interesting that they can start to accumulate phosphorus because why would they want to do that? Why would a bacteria want to pick up phosphate when it can and store it?
Necessary for growth.
Yeah, it's essential, so that's good, but I mean, why not pick up a whole bunch of nitrogen? ATP. So we know the need, so it's economy. You know, we have the demand. I gotta build these, you know, the phospholipids and ATPs and this stuff. Okay, so you gotta think about the supply part. Yeah, it's one thing if you're growing these things in rich media, right, that you pull off the shelf, there's tons of nutrients and phosphates in it. That's great. In nature or in different parts of your body, maybe getting some of these essential nutrients are really, really important. I mean, there's carbon in a lot of places. A lot of carbon you can't metabolize. You know, we know that. There's tons of carbon around us, but, you know, go chew a tree, you're not going to get too far. Oh, it'll be interesting. But the thing is that you kind of make hay while the sun shines if you can do it. So again, you'll see polyphosphates accumulate when there's a chance to encounter them. I think that's enough. If you're interested, at some point we can talk more about one of the most famously studied enzymes is alkaline phosphatase in nature. And I'll leave that for a different day or just on interest, but there's a lot more to this. Okay. Well, there's some polyphosphates. All right, I like these inclusion bodies, sulfur globules. Okay, now here in chromatin, that Fenning took. Love this guy's pictures. I mean, wish we had a gallery of Fenning's pictures. What they do is these guys are picking up sulfide. Okay, hydrogen sulfide is in what form everywhere? Okay, who said chemistry? Who's learned about sulfur? Who learned about sulfide? Learned about sulfide? What form is it in, of the three common states of matter?
Isn't it like in a powder form?
Hmm?
Isn't it like a powder, like a solid form?
Could be, but it's a gas, yeah, hydrogen sulfide, fart gas, right? You guys know about this. You know, it's not uncommon. You know, drive past a pig farm. Sulfur, sulfides, mercaptans, those things are really volatile. In fact, too much of it is toxic. Not just because you're going to pass out because it stinks so bad, but it messes with your cytochrome system. Right? So these guys, they take sulfide. Sulfide is sulfur in its really reduced form. Okay, and they say, reduction. I can get energy off it. There's some hydrogens there, so they do it. Now, a lot of their friends just continue to oxidize it into sulfate. And it goes out as an anion, and that's gone. These guys are kind of smart. They say, well, why not? Again, this is how they talk. If you watch them, listen hard enough. Why don't we take the sulfide, and as we oxidize it, we can make it into a pellet or powder form. A yellow powder, what type of sulfur compound is that? Kind of yellowish. Right? Well, that's sulfur in its elemental form, S0. So they take it and they put it inside of them and store it for a rainy day. You go, now why? Why do that? Because later on, should they need it, they can use that sulfur. It's still, okay, it's more oxidized than sulfide, this is true. But it's not as oxidized as sulfite or sulfate. So they still have reduction power in it. So if you have the mechanisms, they can move it all the way across the different reduction states. Okay. Anyone know what I'm talking about? Okay, give me some, you know what I'm talking about. Okay, you know that. This guy's with me on this. Okay, anyone else? Where did I lose you? Sulfide? Reduced? Okay, it's got an H on it. Negative charge. Well, take a look at that. This is just an introduction to metabolism. We go around oxidation states all over the place. When you learned respiration, it was complicated. Okay, there's citric acid cycle. What's that all about? But it really was glucose going to CO2 and some oxygen and water in there. We substitute oxidation states all along. Part of the next lecture will help you with this. So if you're going kind of lost, it's okay. We'll bring you back to the group and we'll keep going. Now, those of us in lab, which I think are most of you, we started this mud experiment. Why am I paying thousands of dollars in tuition for this guy to get up and make little mud pies? I did that when I was in kindergarten. What a waste of time. This guy's an idiot. Okay, two separate things. Not a waste of time. The guy could be an idiot. But the thing is when we look in there today, perhaps, you'll start to see different things. You'll see you can't see these guys. We have to dig them out. Maybe for the microbial exploration, some of you will dig them out and look for these guys. You can see them at 1000x Fenning. But you will see some areas where there's deposition of elemental sulfur. You'll see the whitish yellow. And some of these guys, this particular guys, will make sulfur and hang on to it for a rainy day. Other, its cousins will take the sulfur and they'll spit it out. Some of the big sulfur deposits in the world were made by bacteria spitting out sulfur, and we can see that in our very own classroom just by using light as our energy source. So these guys have to be phototrophic. So there's an inclusion body for you. These guys are fun. I think we talked about them the first day. Here they are again. Yeah, we did talk about them the first day. And there it is. Magnetic storage inclusion. Using magnetite, basically. They're something that's soluble forms of iron. They can put it into themselves. I don't know the exact mechanism of how they make the inclusion bodies. But they put them in there. Almost looks like a little spinal cord, right? And these guys made them red, so you can't really see them, but they're not red. The magnetosomes are in these guys, and I will say the literature, we had a few students that made, the bacteria is Magnetobacterium, not very hard to remember the genus. We had a few students that went out and devised Magnetobacteria traps, and they went out and started finding these guys. They're pretty common, but the idea that the literature says is that the magnets help them to orient themselves so that they know where they are on the earth, so they can better find where the oxygen is, which, you know, I just never bought that one. It's kind of like, I don't know. But, you know, it's in the literature. We do know that they do key in on the poles, and they key into the opposite pole in the southern hemisphere than they do in the northern hemisphere. I'm pretty sure they key in on the northern pole here, and you know, the south pole, but they do key into different ones depending upon the hemisphere. So there's a lot of mystery behind what these guys actually are doing. Although they have done the genome on this, maybe they know more. Janovich, I think, in Europe, did that a few years ago. Okay, gas vesicles. Here we have cyanobacteria, and it says buoyant. So this is glare, we can't see that, but we see there's kind of a green soupy murk here. And you probably all have seen, especially growing up or being, you know, driving through Illinois, driving past Lake Springfield some days. The thing is we see algal blooms. Algal blooms can either be eukaryotic, so therefore a bunch of plant-like organisms, or protists now, or they can be bacterial, cyanobacteria. Now, one thing that's important on this idea is that this, for me, fits into the idea of active buoyancy. That is, something the organism can do in order to alter its placement in nature. Where passive buoyancy, you kind of are what you are, and you do it, you fall at a certain rate if you're a certain size in physics. But here, some organisms have gas vesicles. Okay, the little spindle rods, and I'm pretty sure the next slide has detail on that. But the point here is if you can affect buoyancy, you know, buoyancy is how you float in the water, you know, life preservers and all that. So they can kind of pump up their little gas vesicles and rise, and then kind of pump them back down to go down in the water. Why would that be useful if you're a cyanobacteria? Now, cyanobacteria, they do photosynthesis one, so they're kind of like plants. Okay, they make oxygen and eat up CO2 during the light. So, but they are bacteria. Why is it helpful to be able to affect how buoyant you are in the water, or basically your position in the water? I'll go, I think you answered last, we'll go with you next and see how this goes.
Because there's more oxygen located at the surface of the water, whereas there's less volume.
Gas. Gas is important. If they're metabolizing, these guys are doing photosynthesis, so, but they can get the CO2 on the top also. Okay, so we have gas exchange, very important, good point.
You can only photosynthesize during daylight, so you may be more buoyant during the daytime, use up that sunlight, that direct sunlight, and then whenever night shifts around, they can no longer undergo photosynthesis, they could sink back down.
Yeah, yes. The concept of light and where you are in position in light. What happens is you go deeper into a lake. You lose light. It gets dark. At the bottom of the lake, it's dark. Okay, I don't know if you've ever done it. It's true. Okay, so if you're just a little particle, I don't care how small you are, you have mass, at some point the Earth is going to pull you down. You're not going to photosynthesize too well. The opposite can be true too. Sometimes you can have photoinhibition through photorespiration. Anybody learn about that? Well, sometimes photosynthesis can be so intense. You know, you learn C3, C4 cam, and then you start, you know, talking about carbon uptake, talk about what photosynthesis, like respiration, if it wasn't controlled, we would melt our cells. Photosynthesis, if done too much, can also inhibit the process. It actually has a feedback loop that's destructive. So the point is, you you can drop down a little bit, also be in the, a good place to get the light rays you need, but, maybe not burn up. In evolution, okay, if we go back about 2 billion years when these guys showed up, and they started spitting oxygen into an anaerobic atmosphere, they had another problem. There was no oxygen, so there was no ozone. So being near the surface, you got fried, quite likely. Great place to have a mutation. Let's go to the surface and mutate. Controlling your buoyancy was selected for over time, over 2 billion years in that also. And of course, now we know they found other homes. The lichens took them in. They don't charge them rent because it's symbiotic. That kind of stuff goes on. So here is a picture of Anabina. Doesn't matter. It's a type of cyanobacteria. It looks like beads, right? Okay. And part of me cares, but I don't care enough to badger you about these names. What I want you to look at is this weaving type of thing. These proteins come together in a structure almost like a little gas sack. A Gore-Tex fabric. It leaves the water out, but lets gas go through. It's kind of like that, except these guys didn't have Gore-Tex to work with, right? So these gas vesicles to move things in and out, and that's how that can work for them. You may see this cell looks different. You think, oh, maybe that one's rotting. We'll talk about that more too. That's a heterocyst. A heterocyst, you can write them now or a week later. We'll talk about it a lot. I just want to let you know we're coming back to it. Heterocysts are able to take nitrogen gas and reduce it, and thus help make proteins and so forth. It's what the symbionts in the soybeans and stuff around here do. So they're like the cyanobacteria. Okay, endospores. Endospores are a propagule. They're highly, highly resistant to like everything. If you heat them up, hard to kill them unless you keep on heating and steaming them. If you put them in the soil, they live for a long time. If they're in your scat, your feces, the endospores don't break down very much. They are a resistant life form of the parent. They in themselves do not reproduce. Okay, it is like, you've learned about spore. You've got to be careful. You've learned about spores of fungi, possibly in general biology. These are a little different. Those spores are formed differently. This is actually a body that's made inside the cell, in most cases, and then the body of the cell dies and goes away, and it leaves this little resistant propagule to take on and weather the storm. Okay. Dormant stage, ideal, yep. Yeah, as far as, I do not know of any gram-negatives that do this, which is interesting. We talk about diversity of the gram-negatives. They don't set that up, okay? Bacillus in the soil, well, in your gut. Clostridium, botulism, those guys, they have this capacity. So the spores can be in different parts of the cell. Now when we talk about the bacteria cell, here we just have a negative stain. This is light microscopy. Looks like Hype did all these, okay? And so the cell itself stains. Now we have to have words, nouns, because we have two types of living matter. So the parent cell is the vegetative cell, like vegetation. It's called the vegetative cell. Okay, the spore is the spore. So when you divide stuff, you gotta make up words. So the vegetative cell here is around these spores eventually will just come out and live, depending on how old the culture are. This is a very young culture. It's been taxed in some way, either by age. Okay, we know in a closed culture, like in lab, they're eventually eating up all the food, and living in their own waste, and eventually the waste becomes somewhat toxic, a feedback loop. It says, well, things aren't going so good, guys. Let's start thinking about making spores. So they did. You can also induce sporulation with manganese. Okay, it turns out that there's an important enzyme that without manganese does not sporulate, but that's just a trick of the trade. Now, these guys, like this is probably Clostridium, I don't think it's listed here. They kind of look like little spoons, okay? Now, it's not like there's nothing there, like, or a needle. It's not like it's a hole there, it's just that spores are so resistant with many, many layers that they don't pick up stain. And again, if you're in lab, we'll do this. If you're not in lab, I'll tell you about it. The spore is so resistant, we have to use a smaller stain, and we have to steam the stain into the spores. We have to kind of open and loosen it up and get that stain to go inside it. It's that dense. Okay, this is just simple staining here. Okay, these guys are almost to the end, and these guys are like bacillus. They're right in the middle. These are fairly incredible shots today. So, but yeah, so here's just our vegetative cell. These guys are dividing, and then we have spores on their own here. So that's what you're seeing there. And you'll, lab people, you'll be seeing that. And this is just giving you what I just talked about so that you have these for your notes. No real new information there. Again, there's names and names and names, and you know, I'd love for us to all sit and memorize everything, but there's just not that much time. My point of this slide is the many walls. The DNA is in there. You can't lose your DNA, okay, if you're a viable type of organism, okay. It does shed some of the cytoplasm, quite a few of the ribosomes, hangs on to a few key enzymes, but really pares itself down, you know, into just a fraction of what the original cell is. And then it grows and induces these different layers, okay? A cortex, a core wall, a spore coating, exosporium. Whatever those things are, they make it hard for you to mess with it. It's like a little bacterial time capsule. Now, you can induce these things to grow, you know, pretty easily. Put them in good media. Then, you know, they get enough of a signal after a time. They'll start saying, "Well, I think it's good. Let's go for it." And they'll start going back to the vegetative type of growth. I only have this in here because it shows the transition of what's happening, so that you kind of see that there's a process to it. But I'm not going. We're going to learn a few different cell cycles. This won't be one of them. This is one of these you mark for my own information. I mean, you're scientists. I know you'd like to know more, so I give it to you. So, some of the differences. I like tables. I don't think tables are there for us to memorize, but they do help us to organize our thoughts. So the appearance, yeah, the vegetative cell does not seem to be as refractal, as hard and unpenetrating as the spore is. Well, we didn't talk too much about calcium. We basically have a metabolizing cell when the vegetative cell is active. These guys aren't, they're hanging out, that's it. Just enough to keep you going, but they're really shut down. I mean, spores would be a perfect thing to put on the next space thing that we send out to goodness knows where. They would do very well in outer space. If you're not metabolizing, you don't need oxygen if you're aerobic, and you're just not doing much, is what this is saying. So, okay, I think you get the idea. Not that big of a deal. And we talked about that. One way to get rid of volume is to get rid of excess water, and that also helps you to stay stable. Water is great, and you need it, okay? And they need some, but water, if it freezes, it makes crystals. Crystals in your cells will be like little razor blades. They'll split them right open, okay? So we have to always think about when we freeze cells. You know, crystals expand. The more water you have, the more you expand. If you have too much inside your little desiccated spore, it's going to, it'll expand and break that coating. So, really adaptively, it's pretty well thought out. And I'm sure the ones that didn't think it out did not survive the eons. Okay, I think we finished up this chapter on locomotion, more active locomotion. Okay. So when we talk about locomotion, the agent or the apparatus of locomotion is the bacteria flagella. Okay. We'll look a little bit more closely at the flagella, but you learned flagella are also in the eukaryotic cells. And when you looked at the flagella of eukaryotic cells, you learn some things about it. You carry out, how do your flagellas look? If you cut them open, what kind of pattern would you see? The 9+2 pattern. You kind of have like a muscle, myosin fibers moving against the central core. So your flagella in general tend to be real whip-like and actually flagellate like a whip. Okay, whether there's just little ones in the back of your throat or your flagella on the back end of a protist, it's more of a flipping motion. Okay, more like the contraction of muscle than we'll learn about here. This will be more of a motorboat rotary action. Also, if we were to cut the flagella here, anywhere in the flagella, it would just look like a rod, okay? It's just a protein filament, okay? Well, why isn't that a fimbriae? Well, the way that it connects and the way that it works makes it not a fimbriae, and we'll talk about that also. So if we were to look at flagella, and this is, I think, one of the most fascinating and unexplored parts of bacteria, because I know they move around more than we give them credit for. If we look at them, we see that different bacteria have different numbers or different arrangements. In the top, we have the polar flagella. I would like you to know these three terms. I don't think I'm asking too much, so if you want to start them. So we have polar, okay. In B, we have a clump. They're called Lithotrichus. What's cool about Lithotrichus and the next one, when they're all around it, Paratrichus, is that they don't get tangled when they move. It's a fairly fascinating study that really doesn't work when you stop the bacteria. You think, oh, they're spinning, they're just going to tangle up. But when you start seeing them move through water, the water is very thick for the flagella, and they stay separated, and they don't hitch up, which is that it doesn't seem like it should make sense, but it does, because it didn't make sense to me, and my students, some of you guys, kept asking the undergrads, and people kept bringing me articles, and we figured it out. So trust me on that. And they're composed of a monomer called flagellin, which is a protein monomer. Did anyone check these out? I forgot to check these in lecture. The other links that I gave you, I always try to check them out. I think they had the making of the flagella. Anyone? So I said, hey, we leave early, you're going to look at those videos, right? Okay, look at those videos. It'll show you the way that they are produced. I'd like you to take a look at that. This is why it's not a pili or a fimbriae, okay, or any of those other A's, okay? This right here, now this is the gram-neg, so let's get our orientation. Okay, let's see, I'm going in the side on this. This is my cell membrane, this is my outer membrane. Oh yeah, there's the LPS, okay, so always take a second, when someone asks you something, you get overwhelmed, you go like, oh, what am I looking at? Take a second, always relate what you know to what you don't know. That's how you do it, and not get flustered. It happens to some of you on tests. Just take a second and look at what you know. Now, the cell membrane is going all around the cell. Let's just keep it simple. There's a polar flagella. Okay. What's interesting on this, and you see the people, I did not know this. I got on YouTube, and people who are into creation science, and there's no judgment in this, I'm just saying that people are using this as an argument for intelligent design. I'm not going anywhere near that argument for or against, but it is a fascinating way of setting stuff up, no matter how it came about. I'm going to stick with evolution because it's a science class. Anyway, in this cell membrane, we have these two rings. Okay, they kind of put themselves into the polar part of the membrane, and we have these flea proteins. Sometimes you say fly, I guess depends on where you grew up. Either way, okay, so we got this kind of switch, and we have these motor proteins called MOT proteins on the outside giving this thing energy. I don't know if you've ever seen like an electric motor but there's big coils on the outside of it that create a magnetic field, and the inside there's an armature, and you have bearings on the end that suspend this thing. You flip the switch, and the motor goes on. This always reminds me of that. I used to build little motors for science class when I was a geeky kid. Now I'm a geeky adult. So now in here we have a peptidoglycan ring, a P ring, that in the gram-neg is their gram-positive also. Now this is about where it stops with the gram-positives. So, but here we have another stabilizing ring up in the lipopolysaccharide area. And so this thing is stabilized in the membrane of the cell. That is really, really cool. Okay, now, here there's a hook. This is just a condensed portion of the protein. And then if you look on YouTube and so forth, what they show you is all those little balls are the little monomers, the flagellin. And this thing will start to assemble and curl around itself and make this long strand on the end. And they vary. Okay, Del Vibrio, it's like 10 lengths of the bacteria itself. And those guys are like Cadillacs because they hunt other bacteria. They're fast. And some flagella are just very, very small. What they all are is, this looks like, you could hit this thing with a sledgehammer and it wouldn't move, but they're really, really fragile. We tend to break them off. Okay, because we're looking at something 14 nanometers, 0.01 micrometer, in width. Whoa, okay, really, really small stuff. That is on the edge of that's smaller than what's visible at 1000x in your microscope. Okay, as we have, in lab, we have to coat the flagella with stains to make it thicker so we can see it. That's how thin these things are. So what happens then is there's a switch in this motor, okay, and it spins, and it's fed ATPs, and I'll give you some story problems. We can work out how many ATPs and how fast this thing spins and see how far these things go, but I won't go into that now. So that's the architecture. Now, I must admit two things. This is, a pretty picture made by an artist, and they did a very nice job. It doesn't look that way, but even if you do an electron micrograph, you can see these structures in there. They're not colored like that or anything. But so here's, it starts its assembly, okay, adds the free proteins, the mots, I see, they're coming around the outside. Then you have extension, okay, through the outer membrane, the hook, and then there's this cap, and then you have additions behind that as this thing takes off. So it takes some time to happen, and it takes energy to both make it and to work it. It's just like the building of any building and the use of a building. Okay, it's a structure and it has that. They think, Costerton out of Montana says, he says, I think bacteria, a lot of them have flagella, and they lose it when they go into a biofilm. But when things get bad, they regrow their flagella, and they'll swim and use that operation. I think he's right, and we've done a little bit of experimentation on that, but I want to tell you that these things, when we start talking about bacteria, there's so much difference. Not everything is just always the same all the time. Let's go on and talk a little bit about that. Let's see, what do I have here? Rotational, okay, yeah, so they can rev up the motor and slow down the motor, okay? How much are you burning? Well, it's not fuel. It's fuel, it's just oxidation reduction fuel, right? It's ATP fuel, it's not, I guess gasoline is oxidation. Anyway, they can rev it up. They can also switch directions. You can turn the motor one way, in which case the flagella is spinning, and with that hook, it gives it, like a propeller, it gives it a cant, so that it can turn it this way, okay, and give you, okay, turn it this way and give you forward motion, and it can reverse it and go backwards motion. More of the polar ones for the reverse, but it does happen with the other ones. Okay. So here we have a Lithotrophus arrangement, but we have the flagella, and it's going what? Well, I guess we're looking at it this way, so it's going counterclockwise, okay, and it's going in a direction, And this is a cool thing, when it's doing this, it's called a run. Not hard, okay, it's moving, it's a run. When it spazzes out like that, it just stops. Now, there's different ideas on this. One is that that's for orientation. Two is that I kind of used up my fuel. I gotta wait a little bit to get some more ATPs over there. Okay. You know, there's different kind of theories what's going on there, and then say, okay, I'm ready to go, and yeah, push upon a clockwise motion, but then it resumes its counterclockwise motion. Okay. Now, when you look at this, we're going to keep building this for a second, but those people who've taken physics go like, well, wait a minute, if I'm spinning, how come I don't make the cell spin? There's got to be rotational vector going into that original cell, right? The deal is the mass. Okay, this thing does tend to want to spin with the cell, okay, but it, no, opposite of the cell is this is spinning the other way, but it's the friction of the cell and the mass that just slows it down relative to the flagella. Okay, so that part's thought of, I believe. Okay, so this is just talking about what we talked about, reversible, can stop and reorient. Now what we want to talk about, oh, and this is bacteria on acid, I guess, or your brain on bacteria, I don't know, but this is just showing that with these guys are funny. They made fluorochromic stains, and they stain a bunch of different cultures, different ways. They put it in here, and then this is a time lapse. But you're getting to see the movement of bacteria. And if we look in lab, we can take out rhodosporidium, and it moves across very fast, but like a big kind of, like a Lincoln Continental, man. You just see it go across, they move. And the other stuff we'll look at moves very fast. Proteus, the wanderer Proteus moves very fast. That's the god of wandering. So I like this for that reason. Let's go back here. Where's my run and tumble? Oh, here it is. I don't know why I put gliding. Let's come back to gliding motility. Let's stay on locomotion and taxis. I don't know why I mixed these up. It's just what happens. But I'm back with you now. So let's talk about movement. Okay, movement for the bacteria. If it's completely random, they'll run in one direction, kind of just do that spaz, what they call a tumble. And as a tumble, when you're moving and stop, you're moving in a fluid, they tend to reorient. And like the one slide says, sometimes they'll put a little counter spin on that, and they'll then go in another direction. And if they just keep doing this kind of randomly, there's no real total movement. They're just kind of checking out their environment. Okay. Some bacteria have taxis. I point to my nose, because in my nose I have chemotaxis in my nose. That's why I do that, okay. But taxis, okay, not, spelled differently than taxes, which, what the government does to us. So, this taxis is like taxi, moving, okay. Chemotaxis is being able to sense a chemical and being able to go to it. So, first off, you have to have the receptor to sense it. It's weird because, we kind of wait for diffusion a lot of times for taxis. Oh, I like that perfume. Who's wearing that? We have a different maybe response, or pheromones, whatever, ants or animals and plants do. These guys are kind of swimming into it, and it's hard to, there's diffusion, but they have to figure out the gradient as they're actually, moving into it, so they can get to the source of it. So, if your runs and tumbles are random, you have no chemotaxis. But in this case, so here it is. These guys either there is no attractive molecule there for them, or they don't have the receptors. That's the only way you can conclude that without knowing more and testing. Okay, let's just say we put an attractant over here. Let's say it's phosphorus, like the video I hope that you will now go look at that Mandy did. So we put phosphorus over here, and this is Pseudomonas. they wanna go find phosphorus, right? We already talked about that today, okay. So what they'll do is that the runs are gonna be more in the direction of the attractant than the runs that they're going away from it, the random ones. So that, so in this case, they're going, okay, pretty good, how's it going here? Here, about the same, getting a little bit less, okay, getting more, okay, more, maybe I go this way, short way, but, so in general, their motion is this vector towards the attractant. Now we break this down. If you look at what the work that Manny did in some of the YouTube videos, this happens fast, okay? You're back there, you don't have time really, well, I wonder if I should go get that attractant. It probably won't be there, so. So if the runs are directional and greater than the time tumbling or the runs away, then you have positive chemotaxis, okay? And we did, we're showing you a live work that the graduate students did, but this shows using a capillary tube that we made and you could make. We just got a flame, and we took pass-through pipettes and we stretch them and cut them, and we try to get a 40 micron opening. Okay. Now I guess Dr. Johnson has a professional or a machine that does this. But we didn't, so we just threw away a lot of pipettes, but we've actually got them. Now, what we have here then is some different things. So here you're putting in, imagine that this is agar or an attractant, that's in agar, something that's just not going to diffuse out necessarily. It's staying somewhat stable around that tip. It's just not going to, going out. Okay, if you put it in there and sorry, this one actually is a solution, it's going up in here. Okay, so this is a solution for the attractant. These guys are kind of evenly distributed. You'd say, well, not a lot of evidence, some swam up in there, that's going to happen. I'm not too convinced. If they start swarming up in there and around where they're getting the attractant, positive chemotaxis. You can test it, phosphate, nitrogen, toxin A, Roundup, whatever you want to try it. If it makes them go away, it's a negative attractant. Well, a negative attractant or a deterrent. Okay, they don't like that. And you go to yourself, well, that's kind of interesting. I like the little dots and the colors. That's kind of cool. But think about it, that these things are actually, they're not just bacteria. They're just, now we know they grow like crazy. But they can move around and find things, and do stuff, like fish and us. So I go, well, but they're just little bitty, yeah. So this is kind of a cool thing to me, because now this little red or blue dot has behavior. I mean, not neurons. Let's not get too carried away, but that is a pretty interesting thing. This is just a graph saying the time. If you're attractant, you're definitely going to see an increase in the cells that are going towards your tube. I don't think that's that. Now, oh yeah, let me go back. So there's different taxes. You can be tactic towards air. We talked about with Magnetobacterium, I'm not convinced. But there are bacteria that will find and grow where the right amount of air is. Oxygen, as you know, is lethal. That's why you have enzymes that get rid of oxides and you have peroxides and super dismutase oxides, because oxygen will go through your body, create free radicals and kill you, okay, or cause cancer and stuff like that. So some bacteria say, I like oxygen, I just don't want that much. I'm going to be microaerophilic. I want a little bit of oxygen, and so forth and so on. So they can show that. Phototaxis. Well, in some ways, buoyancy is kind of phototactic. If there is an actual phototactic response driving it, and not maybe a gas response. So you have to, again, test it. Take the light away, see what they do. Okay. Osmo, salinity. Salinity that isn't specific, so you could test with different salts. Oh, it's not that they really want sodium. That'd be chemotaxis. It's just the amount of halo osmotic pressure I'm giving them causes them to move around. That's possible. And if you're actually attracted to water or not hydrotactic in that case. All right, sorry for the mixing. I know you hate it, I hate it. I'd love to be organized, but sometimes it just takes over. Well, this isn't a great review slide. I'll try to get some review notes up, but you can pretty much guess where we're at. You know, we did contribute to our idea of diversity. I told you, you know, on the test, I'm gonna ask you to draw out and label the cell membrane and outer cell wall and tell me why the Gram stain works. I don't care that you know how many seconds the crystal violet is on the cell. Somebody should know how that mechanism works. I'm telling you, you can go home and write this essay tonight and start studying it. I'm gonna give it to you, you know. We talked about specialized structures, and with the structures, we tried to relate them to function. That's always a theme in biology, or certainly in my course, so if I ask you about that, there's some good stories there. We did take a little bit of a sidestep, and we said, well, not everything is gram-negative. We did have the mycobacteria, the acid-fast guys. Didn't say too much about that. We did talk about the archaea, you know, and then we started talking about locomotion and these other ways of moving the cells around. So, if you're looking for major themes, there they are, and you know, there's a moderate amount of detail. We skipped over some of the very detailed mechanisms. What questions do you have as we forge on into metabolism? How many people is metabolism their favorite subject? Physiology. How about? Really? Okay. When I took it, I love physiology, and I hate it at the same time because you know, the teacher was always. Teachers back then yelled at you. You know, if you didn't get something, what's wrong? Why aren't you getting that the CO2 concentrations causing the turtle to burp? You know, it's like, I don't know. It doesn't make any sense to me. You know, but now we go, well, let's talk. So anyway, I love physiology because it explains how it works. If I had to do things over again, and I was sitting where you were at, I would pick up more prokaryotic physiology because you know it's 95% of the species out there. I think it'd be a good idea to know how they're working. We're going to start out in physiology. We don't have too much time yet, but we're going to start out in physiology, really get down to metabolism. We get a little bit of physiology spread out in the class, but we're going to start out with metabolism. What I hope you know, and you know, I know when I ask you questions, some people just look back at me because that's how you look, and some people are like, I don't know what you're saying. You know, which kind of looks like the first thing. But the thing is if you don't know what respiration is, look up the summary formula of respiration. You know, look up photosynthesis, and make sure you know what's going on. Look through the citric acid cycle. We're going to talk about that. And then we're just talking about a little bit about bacteria differences. What I consider to be a lot of review, which you might say, well, that's not fair. I didn't have that before. Well, I kind of think because we are an upper level course, that you probably saw some of this stuff before on that. Getting into that, let's start out. Aerobic respiration. The summary formula we all learn is what? I guess we don't have erasers, so. All right. Respiration. You're doing it now. What am I doing? We're respiring aerobically. So give me the formula. Or give me what you remember. You know, that's the other way, you know, when someone asks you something, you know, we all feel bad, like, oh, I want to know the answer, I want to be right, I want to get an A. But start out with what you know. You know some part of respiration. I mean, part of it's in the name. There's oxygen in there someplace. Okay, so we got that. Aerobic respiration, I can put an oxygen up here. Okay. What else is with aerobic respiration as we've learned it? Maybe you learned it by a different name, but I think you're getting on even to my language. Go ahead.
So you need a sugar source?
We need a sugar source. And the way most of us learn it in introductory biology, what sugar do I put over here that reacts with this oxygen?
Glucose.
What is glucose?
C6H12.
You are such a great class. Good for you. Good for you. Okay, now get rid of the extracellular matrix, and this is gonna break down into what?
Water.
Carbon dioxide, yes, carbon dioxide and water. And why do we do all this? And we say, well, we gotta balance things because that's the way we are, we're scientists, we like it when things add up. Okay, now, what's going on with the carbon? Here we have carbon that obviously has energy stuck in it, and we gotta get it out of there, okay? And there's a whole great big thing we gotta do, because, we could also, this also works for paper, right? I can get out my Bic lighter and, okay, well, I can't light that, that's some kind of polymer, but I can light this guy's book here. I got aerobic respiration, right? Is it different? What's cellulose? What's the monomer of cellulose? What is where would I find cellulose? Who's ever seen cellulose? Has anyone here seen cellulose?
Yes.
Is it missing?
No.
Go ahead. Where do you see it? Where do you see it? Here's some. Okay. If I look out there, do I see any cellulose? Yeah, it's the most common natural polymer. You can't get out of the bed in the morning and, well, maybe. I mean, it is a pretty synthetic world. But likely you're going to encounter cellulose. What's cellulose made up of? It's starch, but it's a bit different, right? What's starch? Well, regular starch.
Doesn't cellulose have beta linkage base and starch has alpha linkage base?
Yes, it does. Beta and cellulose, alpha and starch. And what's the monomer that's linking together? Hey, glucose. So if I do this, I'm burning paper, right? I'm burning a log in the fireplace. Are you burning a log in your body? In a way, yeah, but you gotta slow it down. That's what metabolism is. You have to have a bunch of reactions because this energy comes out as light and heat. There's some randomness, okay, there's always randomness. But that'll melt you, okay? How do you know? Put your hand in a fire. It's gonna melt and burn and stink and hurt a lot. Okay, don't do that. Imagine that. Okay, but anyway, yes. So we are doing this. We're doing one molecule at a time, and we're breaking down the parts of that molecule. Okay, so carbon has energy in it. In the carbon associated with water. This is a carbohydrate, right? I mean, carbohydrate. So the thing is that carbon reduced or in an oxidized state compared to its product? These are substrates and products. I'm just trying to remind people, I think you already know about it. So is carbon reduced there or is it in an oxidized state? Where's the carbon go? Here. Okay, so is this C more oxidized or less oxidized than this C? What do you think? So let's stick with this one. So this, okay, this one, okay. Is this one more oxidized or less oxidized than the carbons in glucose?
Sorry.
Come on in. You know the answer to this. You've taken this class. Yeah, it's more. Just look, per carbon atom, there's as many oxygens as I can put on there normally. Okay. So these end up over here. So that's the reducing power. What I want you to do, as you look through this chapter, is I want you to look up a definition of respiration. You learn respiration like this. It's messing with you. I want it to set you free. Go back and look up the definition of respiration, or look up aerobic versus anaerobic respiration. Google it, I don't care, wiki it. It doesn't matter to me. You'll find the answer. They're not going to lie to you, and it's not a secret. Okay. But we want to break open that idea starting on Wednesday. Okay, see you then. Well, some of you this afternoon.